Welcome everyone. We're starting week number 5. We're halfway through the term right now, so you only have a few more weeks to go. Let's see what we've got going on. So last week we looked at cellular respiration, which is a way that many organisms on the planet use oxygen. They harness oxygen from the atmosphere to make tons of ATP molecules for energy from different food sources, and in particular glucose. And you took a look at peas and how germinated peas versus ungerminated peas versus beads produced carbon dioxide through the cellular respiration process. So we'll see what you came up with. I hope you had a good experiment. This worked very well for me, so I hope it did for you too. This week's topic is DNA, deoxyribonucleic acid, one of my favorite topics. Here is DNA. I even have a model. Sorry, it's a little bit geeky, but anyway, DNA, the molecule of heredity, and it determines all the characteristics of the organisms that have this particular type of DNA. So it's a tremendously important molecule. It's so important right now in medicine and will be even more in the future in medicine. Personalized medicine is where your doctor will take a look at your DNA and see what types of medicines you can take, what might be beneficial to you, what you have a predisposition for as far as diseases goes. This is going to be used very much in healthcare in the very near future. It's used right now in the process of identifying SARS-CoV-2 virus through the PCR technique, which we're going to talk about more in a minute. But DNA is tremendously important. It tells us so much about who we are because it is who we are. It determines who we are. So we'll talk about this a little bit more, and you'll get into it in your labs also. All right, I want to start this week with your milestone 2 because this is due this week. It's your experimental design. You also need to clarify your hypothesis and make a null hypothesis. All right, before you start this, you need to go back and look at milestone 1 and fix what you need to fix. I wrote a lot on these milestone 1s, so go back and take a look at these because you're going to have to fix these before you move forward. I'm not going to regrade them, but you fix them because they're part of the final project that you will put together. All right, this is the second milestone, where you write out your procedures for your experiment. Make sure you use the correct format, which is outline form, and include all the steps to your experiment, including any replications. Make sure you do all of that. Make sure you fix your hypothesis. There were quite a few hypotheses that I looked at when I did my grading that were not correct, that had some issues with them. So go back and fix that milestone 1 before you do milestone 2. Okay, so milestone 2 is due this week. All right, then the lab that you're going to do this week is cheek cell DNA extraction. So anytime you watch a crime show anymore, you know, they're swabbing the inside of somebody's cheek to collect cheek cells so that they can run a DNA analysis on that person. Well, you're going to do the same procedure, except you're going to chew the inside of your mouth with some sports drink in your mouth so that you can collect a whole bunch of cheek cells so that you can see your DNA. So I've put my pictures of my DNA here so that you can tell what it looks like. So it's sort of stringy, a little bit fibrous, sort of slimy looking, clearish. You'll see that in both the tubes that I have here, the regular size test tube and then the Eppendorf. Actually, that's a PCR tube. The PCR tube that's over there to the right has a little bit of the clearish, sticky looking, slimy looking substance, and that is your DNA. Okay, every crime scene analyst who works on DNA does this type of work. Now, they do it on a smaller scale because they don't want this much DNA, believe it or not. when they're working on DNA analyses. But they do really virtually the same type of procedure. So I want to mention a couple of things in this lab. CC stands for cubic centimeter, which is equal to 1 milliliter on your small cup. So if you ever see CC, it means mill. That's all it means. Your ethanol, here is the tip of the week. Your ethanol has to be extremely cold in order to precipitate out the DNA. And this means that you need to put it in the freezer. So as soon as you finish watching this video, you go put that bottle of ethanol into your freezer so that you have it ready for when you do that experiment. It needs to be extremely cold, and if it is, the DNA will precipitate out of that solution very well and very quickly, and it will be easy to do. If it's not, if it's warm, it is not going to work. I'll tell you that right now. It has to be cold. This is a quick lab, so realize that. When I looked at the write-up, it says it's going to take you 50 minutes to actually do the lab after you set it up. 50 minutes? How's it going to take you that long? Anyway, no, it's not. It's a fairly quick lab, as long as you do everything correctly. All right, and then also you're going to do a PCR simulation through the link that you're given in Module 5, and you're going to answer the questions that have to do with the process. All right, I want to go through the process for just 1 minute. There are 3 basic steps to PCR. PCR stands for the polymerase chain reaction. Well, what the heck does that mean? Chain reaction means that it's going to happen over and over and over again, right? Polymerase is an enzyme, ASE, that gives you a clue, is an enzyme that makes a polymer. And polymer just means many subunits. That's all it means. So in this case, we're going to use DNA polymerase to make copies of your target DNA that you have put into the tube. There are 3 steps, basic steps to this process. The denaturation step, denature. The 2 strands of DNA break apart from each other. Remember the double helix here? So each of these strands will be separated from the other one. These bonds right here between the bases break. And that's because you're going to raise the DNA to a very high temperature, almost the boiling point of water. When you do that, you break apart the 2 backbones of the DNA. And that's important because you need to get to the information that's contained in the bases of the DNA. Okay, the annealing step is next. Anneal just means to bind to or attach to. That's all that means. So the annealing step is where primers, usually of RNA, attached to either side of the target sequence of DNA. One on one strand, one on the other strand. There you go. Like this. So the primer's attached to either side of the target area. That's because DNA polymerase, this enzyme that makes new DNA, has to have something to get started with. It needs a primer. So if you've ever cut grass before and used a lawnmower, you know that you usually have to press the primer button to give the engine a little bit extra gas to get it going, right? So that's what a primer is. It gets this process started. That's the annealing step. And then finally, we have the amplification step where the new DNA is being made by DNA polymerase. Amplify. You're increasing, right? You're increasing the number of DNA molecules. So what the heck is the purpose of this technique? The purpose of it is to take even as little as 1 piece of DNA and making many copies, billions of copies even, of the DNA. And then you can actually look at it. You can sequence it. You can examine it in all different ways. There are lots of ways. To see what kind of DNA that you have there. This process is what is being used right now for the COVID cases. If you go and get tested, there are 2 different types of tests that you can get. One is the PCR, and it really is called RT-PCR. It's reverse transcriptase PCR, because the COVID virus or the coronavirus that's going around, SARS-CoV-2, is an RNA virus. So you start out with viral RNA, You make some DNA from it, and then you copy the DNA over and over and over and over and over again to see if the viral DNA is actually there. That's one of the steps, that's one of the tests that's used right now to test for the virus. So that's going to tell you if you've been infected, if you are infected with the virus right now. The other type of test that you can have run is the antibody test, and that tells you whether your immune system has reacted to the virus in your body. And so, have you made these proteins called antibodies because you've been exposed to the virus? This tells you if you have ever been exposed to the virus. And you continue to have these antibodies in your bloodstream for hopefully a long time, let's hope, if you have ever been exposed. So that's the second type of test that you can have. But PCR is one. So we're using this test right now to check to see if you have the coronavirus in you. So that's PCR, denature, anneal, amplify. All of these steps have to do with heating. So the denaturation step is very hot. Annealing is a much lower temperature, around 50 degrees Celsius. Amplification by the DNA polymerase is around 72 degrees C. So there are different heating steps in this technique. And so you use something called a thermal cycler, which just heats and cools tubes very quickly in order to carry out PCR. That's basically what you need. All right, and then one word about next week. Next week is going to take you 8 days to grow the seeds. You grow 2 sets of seeds. Each set of seeds is going to take 4 days. If you only grow them for 3 days, you're not going to see the characteristics very well. So you literally need 8 days to grow the seeds. Well, there are only 7 days in a week, as I recall, right? So what I suggest is that toward the end of this week, like Thursday or Friday, start growing your seeds for next week. That will make everything a whole lot easier. Just follow the directions. It doesn't take much time. You're taking seeds, you're putting them in a petri dish with a little bit of water on a filter paper. The setup is not difficult. Put the seeds under a light source. Don't put these under an incandescent light. Put them under either an LED or a fluorescent light, but LEDs are perfectly fine. They don't get too hot. And start growing your seeds. For 4 days, you're basically doing nothing but making sure that everything's okay with your little petri dishes. And then you start over with another set. Does not take long to set them up. Grow them for another 4 days. Look at them, record the results that you got. So please, please, please make sure you start these seeds by the end of this week. I would say Thursday or Friday at the latest, so that you have enough time at the end of next week to write up your lab report. And as always, if you need anything, if you have any questions, please email me at m.sigmon@snhu.edu. I am here for you. I will help you do whatever you need me to help you with. I promise I can help you get unstuck. I can help you with calculations. I can help you with problems. We'll work on this however we need to. We can do a WebEx meeting. We can do it through email, through a phone call. However you want to do it, we can do it. Okay, so let me know if you need anything, and have a wonderful week.